Welcome everyone. Thank you for joining us for our first distinguished lecture of this academic year, and indeed our first virtual distinguished lecture since the arrival of COVID-19. We have 1,000 people registered, and I extend a very warm welcome to all of you, our staff, students, alumni and supporters from around the world. For those of you who don't know me, my name is Mark Spearing and I'm the Vice President for Research and Enterprise here at the University of Southampton. You may not be aware that Southampton is a world leader in respiratory medicine, from developing potential vaccines to leading on national policy. During the pandemic, our supporter community raised nearly £700,000 for the Southampton Coronavirus Response Fund. These generous donations supported our students and cutting-edge medical research, including our respiratory research. Thank you to all those who donated, many of whom are joining us tonight. We're delighted that Professor Stephen Holgate is joining us this evening to deliver his lecture, Understanding and Living with COVID-19, Past, Present and Future, ? A little bit of introduction of Stephen, who really needs no introduction, but since joining Southampton in 1980, Stephen has transformed our understanding of respiratory diseases. His research has informed guidelines at the highest level, as evident in his involvement in a recent government report written with 37 other experts on how the pandemic will develop this winter. Stephen is a Medical Research Council clinical professor of immunopharmacology and honorary consultant physician at the University of Southampton. He is recognized as one of the top specialists in his field nationally and internationally. He has devoted his career focusing on the interface between basic sciences and clinical application to improve our understanding and treatment of allergy and asthma. And he remains one of the world's foremost spokespeople on the dangerous impact of air pollution. He is also one of the co-founders of Synergen, the University of Southampton spin-out company established in 2004 to develop inhaled interferon beta, which has proven effective against viral respiratory diseases, including now COVID-19. And on behalf of the university, I'd also like to pass on my sincere congratulations to Stephen, who was knighted for services to medical research in the 2020 Queen's Birthday Honours List just last week. So, for the first time in public, Sir Stephen, over to you.
Well, thank you very much indeed, Mark. Ladies and gentlemen, thank you very much for joining me this evening in this discussion. And I'm sure COVID-19 has been at the forefront of your minds for the last 6 months. So what I hope to do in my talk today is to give you some possible future glint that there may be something very positive that's going to come out of all of this at the end of the day. And despite all the gloom and doom, things are moving on in a very satisfactory way, even though we're having to struggle with it in our lives at the current time. So let me share my screen, which I will do now. And there we go. And let me begin the talk. There we go. That's it. Good. Thank you. So in picking the title of my talk this evening, I wanted really to give a great sense of optimism really as we go forward, because this last 6 months, there's never been anything like it in most of our lives ever before. And I think we have lessons to learn as we look back and then look forward, hence the title Past, Present and Future. But let me begin at the beginning. And of course, this is a virus that we're talking about here that's rather unusual. I mean, viruses, generally speaking, are designed just to reproduce. That's what their main function is just to reproduce. And their whole machinery is designed at reproducing. So if they can get into the body and into cells within particular tissues, their job is to keep reproducing themselves. And as you'll see on the right-hand side, nobody's spared this. And obviously, there are no customers that are not going to get this virus unless there are particular protective mechanisms that are in place. And I want to talk about some of those tonight because I think they are really important. Let me start at the beginning, because coronaviruses were discovered really very close to Southampton. David Tyrrell, shown here in this picture, ran a unit in Salisbury called the Common Cold Unit, and it was funded by the Medical Research Council. And in 1964 and 1965, working with June Almeida in London, they discovered this peculiar virus that seemed to cause common colds and sore throats. And you can see an electron micrograph picture of the virus with its spikes sticking out, hence its name, coronavirus. And this particular virus seemed to be associated with a rather severe common cold, but that really was where it stopped. So let's have a little look now at what's happened since then. In the 1960s, 4 different types of coronavirus linked to common colds were described, and they're listed on the left there. But then suddenly in 2002, we had this terrible disease that broke out in China, severe adult respiratory syndrome, SARS-CoV. And this resulted in over 770 deaths. And you remember that because Hong Kong shut down during that period, and that was a particularly bad period. But it didn't really spread much further beyond Southeast Asia. And then we had a second outbreak in 2013 of another coronavirus. This is the Middle Eastern respiratory syndrome coronavirus. And this resulted in just over 850 deaths. And this again was restricted largely to the Middle East, though spread a little wider into some parts of Europe. And as you will know, it fizzled out eventually and disappeared. And then suddenly, what happens? We get this virus, this coronavirus class emerging again in a dramatic manner at the beginning of this year. In fact, if we just have a little look at these viruses as to where they might come from, there are some common features which I think might interest you. First of all, many of these viruses, including the SARS viruses, as well as rabies and Ebola, originate in bats. And that's no accident, because bats have developed to be able to act as a reservoir for many of these so-called zoonotic viruses, viruses that can move from one animal into the human being. And they are the host for a great range of different human disease-causing viruses. Listed above are a few of them there. So why don't bats get the disease themselves? You would think with all these viruses circulating in their blood that they would be ill most of the time. Well, they've developed a special form of immune response called the innate antiviral protection response by generating interferon. And interferons are molecules that actually neutralize viruses in quite a complex way, which I'll try and explain as we go through the talk. Now, the reason why bats don't die of all of these viruses is that the cells adjacent to those that are infected somehow develop a protective mechanism such that the virus can't infect those cells, and the interferon helps those cells from being infected. In other words, the interferon's allowing the virus to persist in the bat without actually manifesting in disease, because they make so much of this particular material. And the reason why it's escaped into the human is because we've disturbed the environment, the bat's habitat, and stressed the animals, and they shed much more virus when they are stressed, both in their saliva and urine, as well as their feces, and can infect other animals. And this increases, of course, their ability to move into the human, where when they reach the human, there is this increased virulence and infectivity and havoc, as we've witnessed with this particular outbreak. So on the left here is a description really of the history of what's happened with this particular coronavirus. And SARS-CoV-2, as it's come to be called, shown in the middle diagram here, in this one here, the Wuhan virus, as you can see here, belongs to a unique class of coronaviruses, very different from the right-hand side here, that the common cold viruses reside in. So these are genetic variants of these viruses. And this particular group on the left here, in which the SARS-CoV exists as well, are transmitted into various animals. And we don't really know how it got from the bat into the human, but it's suspected that there is an intermediate vector here. This pangolin has been suspected as the vector. And these were sold in the open meat markets in China, which you can see here, where the original virus was traced back to. And of course, if you recall, the original SARS-CoV, the one that broke out in China and moved to Hong Kong, that also came out of a meat market where civet cats were infected and led to the breakout into the human race. So this is the history since the end of December 2019. And you can see how rapidly things developed from the first reported case all the way through, right the way now to the COVID-19 disease becoming an international pandemic and SARS-CoV-2 being named in February of this year. And the lockdown, obviously, in China on the 23rd of January 2020 signaled the real importance of this disease and the reason why the global health emergency was declared on the 30th of January, which we're all very aware of. So all of this happened extremely quickly as it spread around the world. And this is the situation yesterday afternoon. This is the Johns Hopkins dashboard looking at coronavirus infection across the world. And you can see at present we're at 38,215,510 cases with over 1,087,000 deaths. I mean, this really is a pandemic plus. And you can see the countries that are particularly affected. Europe, obviously, the Americas, India. Interestingly, Africa still quite well protected. And some of the outlying regions also relatively protected at the moment. This is where we are in the United Kingdom at present. This is of yesterday afternoon on October the 14th, with 15,000 cases currently having been reported, and a total number of cases of 635. So far, we've experienced just over 43,000 deaths in this country. But what is concerning, as you can see at the top right-hand side of this slide, is that we've got more than 100 deaths occurring in 24 hours in the last day or two. And you can see in that map where a lot of the activity with this virus is happening, mostly in the north of England and in Northern Ireland and Scotland at the moment, as things are changing extremely rapidly. So this is really a problem, and this idea that we're coming into a second wave is obviously catching hold. Now, before I go much further, I wanted just to explain one or two basic principles that I'm going to touch on. Firstly, how the virus gets into the human once it leaves its animal vector. And here we can see that the coronavirus has these coronas around it, which possess a protein called the spike protein. And this spike protein here is picked up by a receptor, which we all have on our epithelial and vascular lining cells, called the angiotensin converting enzyme 2 receptor. And ACE2 is an enzyme which in our normal functioning cuts a small amino acid peptide and releases a vasodilator peptide, helping to protect us from factors that drive hypertension. So it has a very important normal function in body homeostasis. But this virus has utilized this as a little door to get into the human cells. The second thing I want to talk about is what happens when the virus gets inside a human cell. And I talked about interferons in bats, but of course we can make interferons as well. And when the virus gets inside the human cell, then it generates the interferons. And a number of them have been described as shown on the right-hand side of this slide. The ones I'm going to talk about mostly are interferon alpha and beta, but they all have very much similar functions and being very strongly antiviral. So those are the two basic principles I want to build my talk around this evening. But let's look at COVID-19 itself. This is a massive study called Open Safely, which is the National Health Service record study of over 17.5 million patients, looking at the risk factors that lead to COVID-19. And you can see the male is very strong. Older age, as seen on the left-hand side, particularly as you pass through beyond the age of 59 into the older age group. Deprivation, certain diseases like diabetes, obesity, smoking-related chronic obstructive lung disease, severe asthma, other medical conditions such as those receiving immunosuppressants for transplantation, And of course, a very interesting thing that's emerged, which is ethnicity, that the Black and Asian people seem to be at increased risk of developing COVID-19 and becoming infected with this virus, which may have some quite interesting and important consequences. So I say past because past is interesting. What's happened is that it appears we inherited a bunch of genes from some of our Neanderthal ancestors. Some 60,000 years ago, Homo sapiens started to interact with Neanderthals and shared their children. And this is when we managed to inherit a set of genes from this particular group of animals or humans, whatever you would like to call them at that time. And in a study of nearly 3,200 patients conducted in Edinburgh, we can see that looking at the human genome, and shown in the middle here are the 22 chromosomes of the human genome. And what's been done here in this Manhattan plot is to look at variants across each chromosome, all 22 of them, to find out which variants seem to link with the development of severe COVID-19, hospitalized patients in this case. And you can see on the left-hand side here in the red dots that a number of these variants statistically very closely linked to genes sitting in this chromosome 3 region. And in this particular region, there is, of course, a very important set of molecules that are encoded that could encourage the COVID virus to replicate within cells, particularly one molecule which enhances the ability of the virus to use the ACE2 receptor to get inside of the cell, and a second bunch of genes which are involved in the inflammation, enhancing the inflammation that COVID-19 causes. So really interesting historical perspective here. Even more so when we look at the distribution of this particular genetic variants, we can see that people in Southeast Asia have far greater prevalence of this particular genetic inheritance than people in Europe. And it might go part of the way to suggest why some people who are of South Asian origin may be at increased risk of developing severe COVID-19. Well, genetics is going to be important here because we know that a lot of people survive this virus and are asymptomatic. So we've got to try and understand what it is about those that do survive from those that get severe disease. And there's a big genetic study funded by the Medical Research Council and by the Wellcome Trust set up in Edinburgh is running at the moment, trying to look at other genetic variants in the virus and in the human that might explain why some patients are so much at risk. And please do look up at the website if you're interested in that. So let me go back to the infection process. Just like the common cold, this virus gets into the nose, shown on the left-hand side in cross-section, and infects the respiratory epithelium in the nasal passages. But it doesn't lead to the characteristic cold that you and I commonly experiencing, the sneeze and the sore throat and the runny nose. No, it doesn't. It silently occupies the epithelium until it reaches the olfactory epithelium, the epithelium overlying the nerves, which are shown on the bottom left-hand side, that give us the smell and taste that we are very familiar with. Of course, one of the characteristic symptoms of COVID-19 early in its development is the loss of taste and smell. And this is because, as you will see on the top right-hand side, that the ACE2 receptor, this receptor for the virus, is very much higher in frequency in the epithelium, the lining cells, over those olfactory nerves. And therefore, those particular cells are going to get a lot more infected with this virus than the rest of the epithelium in the nose. And you can see in the bottom right-hand side that the numbers of cells and the concentration of this receptor is very much higher in that so-called olfactory epithelium. Well, the next thing that happens, of course, is that the virus, when it gets inside the epithelial cell, gallops down into the trachea, the big breathing tube in your throat, moves down the 27 branches of the airways until it reaches the air sacs, where we have oxygen and carbon dioxide exchange, the so-called alveoli, the little air sacs at the very end of the lung. And if the virus reaches these cells, then it infects what we call the type 2 pneumocyte, which is the epithelial cell in the alveoli that secretes a substance called surfactant that keeps the alveoli open. And if it gets inside those cells, then it can switch on the interferon pathway, and this activates the inflammatory cascade. And this involves two major cell types. One's called a macrophage, and the other's called a T lymphocyte. And both these cells are involved in protective immunity. And those that don't progress much beyond this point, these cells fight the virus very effectively, and that's the end of it, and they don't get the disease. But should it actually escape their protection, then we go into this very unpleasant second phase. This usually happens after about 3 to 4 days, and the alveoli, as you can see on the right there, become inflamed, and we get this so-called hyaline membrane formed with proteins deposited around the alveoli, around the air sacs, which unfortunately prevent oxygen from getting across from the breathed air into the bloodstream, giving rise to the difficult breathing problems that we have. And this is why it's called an adult respiratory syndrome, because it's this blockage of the transfer of oxygen into the blood that causes the major problem. The other thing this virus does is make us have little hemorrhages, shown in the bottom left-hand side, in the blood vessels surrounding the alveoli. And then we get little clots formed in the small blood vessels, which slow down the blood and increase the chances of developing thrombosis. So this is getting quite complicated now, a combination of inflammation and an increased clotting of the blood, increasing the chance of big blood clots forming elsewhere. So let's go step by step through what happens. Here's the virus on the left-hand side with its spike protein that you can see. You can see the ACE2 receptor and the spike protein engaging with the ACE2 receptor. We have an enzyme which cuts off a little bit of the spike protein, and that eases the virus inside the lining epithelial cell, first in the nose and then down in the lower airways. And in the bottom left-hand side, you can see that once the virus gets inside the cell, it hijacks the human protein manufacturing apparatus and turns it from making human proteins into making viral proteins and more virus. In other words, the virus is now stealing the human reproduction system inside the cell to make it more of itself, and it reproduces itself. And to stop that, of course, what happens is that these epithelial cells start making this interferon, just like the bats do. Unfortunately, we don't make as much interferon as the bats do, but we make some, and those that are able to make quite a bit of it, such as young children and babies, they do not develop such severe disease and can neutralize the virus quite effectively. And they neutralize the virus because by making interferon, the cells that become infective melt away, or alternatively, these macrophage cells that I referred to earlier come up and gobble up the epithelial cell so it doesn't cause any further damage. And that happens within the first 3 days of infection. If that fails and the virus moves on, we go into this next stage. And this is where we start getting into a difficult situation. This next stage is all about activating what we call the adaptive immune system. This is our immune response as we currently understand it. When we become immune to something, we talk about the adaptive immune response. When we're immune to tuberculosis, it's the adaptive immune response that protects us. And this again involves the interferons, which primes the special cells, firstly that lead to antibody formation, as you can see here, which kills the virus and blocks the virus from doing things. And secondly, activates those T lymphocytes I talked about to produce more killing of the infected cells. So this is an important part of our immune response. So what has happened with coronavirus that causes this terrible disease? Well, there's been some trickery going on inside of the bats, because what's happened here is that the virus has adapted itself to these immune responses. It's trying to evade antibodies by putting lots of sugars around its spike protein, so antibodies can't gain access and neutralize the virus. In terms of inducing this interferon protein, which is so important in protection, we have this sensor protein as the virus gets inside the cell, which triggers the infected epithelial cell to make this interferon beta, which then generates the antiviral protection in all the rest of the cells down the airway. But what this virus has done here is to actually hijack some of this apparatus. So when the virus gets inside the cell here, what the SARS-CoV-2 does is that it blocks the sensor protein, so it no longer sees the virus, and also blocks the mechanism whereby the human cell can actually make interferon. So at these two sites here are blocking the ability of the cell to be able to neutralize the virus. And meantime, the virus keeps on replicating. So the reason why this is such a horrible virus is that it evades this first step of the innate immune response. Now, as I mentioned before, if it does do that, then we do have the adaptive immune response, but that takes several days to lock in. And we hope it does lock in, because obviously we want to get antibodies and lymphocytes to neutralize the virus problem. But the difficulty here is that people differ in their ability to be able to mount this adaptive immune response. And of course, we've been hearing one or two stories of people receiving second COVID infections where they've been quite seriously ill. So the first infection hasn't protected them against the second. But nevertheless, it is possible to try and harness this adaptive immune response to make antibodies and make T lymphocytes attack the virus in the form of a vaccine. And a number of these vaccines are being developed, as you're all aware of. The one that's moving most rapidly is the one which has got the spike protein expressed inside the safe virus, the one that AstraZeneca is developing with the Oxford team. This is it here, AZD1222. As you know, this has gone through safety studies and is now being tested in the United States and in this country. And we may be hearing more about that before too long. So really quite an optimistic sign coming here. We've got good antibody production. We don't know yet whether the virus is neutralized by those antibodies, but it's looking pretty good at this stage. So let's keep our fingers crossed. So the report that Mark referred to earlier on was the report I was involved with the Academy of Medical Sciences, because we knew really back in late May that we might be heading for quite a problem this winter. And a resurgence of this virus, of course, is what we're worried about. And what we're witnessing now, of course, is a resurgence. And when we had this meetings, which took place over a period of 6 weeks through May, June, and early July, we were able to come up with a mathematical prediction that we would be entering a second phase. And this is shown here with this R value. The worst case scenario, we said it would be an R value of 1.7. Less so at 1.5. We're currently at about 1.2 here in the UK on average. There's been 100 deaths so far in 24 hours, so you can see where that red line is. We're a little bit ahead of what the predictions are at the moment, and this is obviously very concerning as we go forward. And certainly we have to see things change if we're going to stop this from spreading. The important thing here is that our report really was to try and help the government and help people arm themselves across the summer, ready to cope with these particular rises in infection rate. Now, whether or not we've managed to achieve that, I can't tell you. We advise greater public information. We advise reorganizing health and social care to maintain COVID-19 separate places away from normal treatment. The adequate PPE and the COVID-19 free zone, absolutely crucial and linked to the test and trace system, which we're all now very familiar with. The capacity to roll out this test and trace system into the most important parts of society was another key recommendation. And real-time population surveillance as is taking place in Southampton at the moment, is what we recommended as well. And vaccination of a larger number of people against the influenza virus, which is obviously just about to take place, is what we also recommended. Now, whether these things you think have been adequately done is up to you. I think we have all been surprised at the rapidity of the resurgence of this virus. And it's not just in Britain here, it's in France and Germany and all the other countries across Europe and the rest of the world. So we really have a problem in front of us, and we desperately need some better treatments and better ways of coping with this. Now, this shows a study demonstrating the time course of COVID-19. And it demonstrates that most people who develop this infection recover quite quickly, within 10 to 15 days on average. But you'll see on the right-hand side of this histogram that there are people who are still symptomatic 35 or 40 days after being infected. So these are the sort of people who are developing atypical symptoms now, and something that's been called long COVID. You'll have been reading about that in the papers a bit today if you've had chance. And these are people experiencing symptoms which did not come with the original virus infection, but are coming subsequently. And this long COVID is a new disease in a way where people really haven't got any experience of it before. And if you're interested in it, there is a website you can go into, set up by Zoe there, which I've put up on this slide, where you can find out more about long COVID and enroll, if you would like to, in their studies. But in essence, what we're talking about here is a multi-system disorder. Shown at the top there is a description of somebody suffering from long COVID, a young person at University College in London who describes it perfectly, that they really were completely taken off their feet, going from one day to another, not knowing whether they're going to be able to do anything that they normally did. And this may last days, weeks, or months. And on the right-hand side is a description in the New York Times where you can see here the genuine concern that this is actually a new medical problem that's emerging that the National Health Service is going to have to cope with. You can see in the pictorial diagram in the center there, the various components of this. There are now 16 different symptoms that have been described in long COVID. But I think what's looking increasingly likely is that there are particular features that have commonality with chronic fatigue syndrome and ME, which of course can occur after virus infections. And here you see the insomnia, the fatigue, particularly the post-exercise fatigue, the headache, the brain fog, characteristic of chronic fatigue syndrome. So we've got a lot to learn about this. Thank goodness the NHS now has invested money in clinics that are going to look after and help these patients, because they're going to need it going forward. And the other important thing to remember here is that this isn't just about hospital patients, it's about patients who also have mild disease. And there is a large study now set up, being coordinated from Leicester, to follow up over 3,000 patients who have been hospitalized for COVID-19, to look at the long-term consequences and to try and understand more about the biology of long COVID. And again, if you want to look up that website, do go in and have a look at it. There's a lot of information about it on that. So what about those who don't develop COVID, who are the so-called asymptomatic carriers? Well, you can see in these CT diagrams that they may be asymptomatic, but in those little red arrows that you can see in the lung tissue, there are little pockets of infection. And in fact, asymptomatic carriers may in fact be infected, but limiting the infection very locally inside small areas of the lung, and therefore do not develop the systemic symptoms we understand as COVID-19. Now, why is it that some people go on to develop this unpleasant side of this virus infection? And I want to get back to this type 1 interferon story again, because there's some genetic studies that have shown that those that get severe COVID-19 have a defect in their ability to generate this interferon. Secondly, in those that have got infection with this virus and are studied, the pathology that's studied, the signature in the lung when they're going on to develop this severe disease lacks this interferon response, i.e. they are not mounting this first step in the innate immune response. and they are not generating their interferons adequately. And this is what happens in older people, those with diabetes or obese, those with COPD and asthma, and in immunocompromised people. Their ability to generate these interferons is compromised, hence their risk is increased. And just taking that one step further, this is meant to be an epithelial cell here, and here we have the virus coming into the cell here, being sensed by the sensor proteins, which become changed in terms of activating other proteins in the cell that trigger the epithelial cell to make these type 1 interferons. And these type 1 interferons then are released and act on the normal cell through a common receptor here and make this normal cell protected against virus infection by switching on over 1,000 different genes within that normal cell so that the cell is not infected with the virus and is able to resist it, hence acting like the bat in resisting the virus infection. Well, in terms of where we're going, inborn errors of metabolism of this pathway, i.e. genetic abnormalities in these sensing proteins and in the transmission of that sensing into the nucleus, is increasing the risk of patients developing serious COVID-19 pneumonia. And again, it's pointing to an increased risk factor of developing this disease. Other genetic studies, this is another big one that's coming out very soon, looking at what we call a whole genome survey, similar to the one I've just described in the primitive people, where you can see that antiviral genes, particularly those involved in protecting us against COVID, are impaired in their production, and that there are genetic abnormalities in the system in patients who are at increased risk of this disease. Furthermore, a very recent study published only a few days ago showed that in nearly 1,000 patients with life-threatening COVID-19, about 10% of them develop antibodies that neutralize some of these interferons, but fortunately not interferon beta, but some of the other interferons, and that this is associated with more severe disease. So as I now go forward as to how Southampton and we got involved in all of this, I bring us back to David Tyrrell and Salisbury. Remember, David discovered the coronaviruses back in the 1960s at the Common Cold Centre, which was an old American RAF hospital in Salisbury, as you can see an aerial picture of it there. And he used to have volunteers that you can see in the bottom left here and infect them with these common cold viruses and have them stay in these Nissen huts for several weeks while he studied what viruses did. And I was very lucky to meet this man when I was a young medical registrar working at Salisbury Infirmary. And when I came to Southampton, one of my research fellows, Sebastian Johnson, shown in this picture, worked with David Tyrrell and his team and developed the first diagnostic test for the common cold virus, the human rhinovirus. And that enabled us really to start beginning to study the common cold virus as a cause of worsening asthma in children. And as you can see, a picture here of me looking a little bit younger, this time with the late Princess Diana, who unfortunately died, in a study funded by the British Lung Foundation in 108 children, which enabled us to demonstrate in those children, and 2 of them are shown here, that whenever they developed a bad attack of asthma lasting several days, the common cold virus preceded those nasty attacks. So this was an important study. And we went on to show that the epithelium, just as with the coronavirus, the epithelium in the lung became infected with these viruses. And that led us really to start looking at why asthmatic and other respiratory disease patients were so vulnerable to these common cold viruses. And there are many hundreds of different types of rhinovirus shown in the top part there, and they use a different set of receptors than the coronavirus, and they're shown in the bottom, 3 different receptor subtypes. And here you can see the experiments that my colleagues did in Southampton all those years ago now, showing that normal epithelial cells, when infected with the common cold virus, could deal with the virus very effectively through the interferon pathway. But if you had asthma, particularly severe asthma or COPD, you had an impaired ability to generate interferons, and that led to the worsening asthma. So that led us to patenting that discovery and setting up the company Synergen in 2004. as Mark Spearing very kindly said in my introduction. And we started studying inhaled interferon in asthma and COPD. And we were able to use the inhaled interferon and measure cells in the airways, in the sputum, and demonstrate, as you can see in the orange bars there, that these genes that are associated with stimulating the interferon regulatory pathways, the so-called interferon protective genes, were all upregulated. There's only 4 of them, and we could have measured hundreds of them to demonstrate that by giving inhaled interferon to asthmatics and COPD, we could produce this amazing protection in the lung locally, in both diseases. And of course, this is what really led us into a clinical trial. And here we had this virus that was evading the interferon pathway. And here we had a treatment in the box, so as to speak, that could put the interferon back. So we set up a study, as you will see here, and I want to thank Tom Wilkinson at Southampton and my friends at the Synergen company, Richard Marsden and Philip Monk in particular, who really worked tirelessly to get this trial funded and delivered. A placebo-controlled trial, 50 patients treated with the drug, 50 patients on placebo, studied over 28 days, having the treatment once a day for 14 days. And the results were dramatic. They truly were. So this is the intention to treat, and this is looking at patients completely recovering from the COVID-19 in hospitalized patients. These are patients admitted to hospital with the disease. And you can see here, if you look at the blue symbols, the intention to treat population, and those that required oxygen, as you can see here, almost improved twice as quickly back to normal than those that had normal treatment. And if we look at symptoms, and this is just one of them here, shortness of breath, again, you can see the drug had a dramatic effect in reducing the symptoms of breathlessness. And these results really were the best clinical results of any clinical trial so far of an anti-COVID treatment, because they reduced the odds of developing severe disease by nearly 80%. They increased the likelihood of recovery, and that means complete recovery, 2 to 3-fold. And they affected the symptoms in a major way like breathlessness. So terrific stuff. And we've got to go now and repeat all of this in much bigger studies. So looking forwards, I have a lot to thank Southampton for, because it gave me the opportunity to be able to, with my colleagues, do some of this work. Yes, we do need a vaccine. It's important that we increase our adaptive immunity and protect the population. But we also need drugs that obviously stop the virus in its tracks once people are infected. And we do not have a drug like that available to us yet. And hopefully interferon might be. We need to understand the long-term effects of this terrible virus infection. And I'd like to suggest that this interferon journey that we've been on, that we've learned something from the bats about how the human race might be able to protect itself in the long term by switching on the innate immune response and not just relying on vaccines to give us the adaptive response. So I think working together, we can beat this wretched virus. So let's get to it and see if we can come out this winter in a positive mode. Thank you all very much.
Many, many thanks, Stephen. Many thanks, Stephen, for a very clear and engaging talk, and it's been fascinating to hear from you this evening. I'd like to also thank everyone for joining us tonight, for your interest in this topic and your continuing support of the University of Southampton. And Stephen, you should imagine the applause of 1,000 people right now, but sadly the technology doesn't allow that to convey us. And with that, thank you all very much and have a great remainder of your evening. Take care.